Okay, good day all. Today we are going to look at metabolic regulation. So the principles of metabolic regulation, we are starting with in fact, let's start with what is the metabolism itself. When we said metabolism, it means the total reactions, or is the sum total of all the biological reactions that are taking place in the body. So whenever we said metabolisms, generally we know that there are reactions that are taking place in the living system and also outside the living system. So those sum of all the biological reactions that are taking place in the living system, we call it metabolism. So that is the meaning of metabolism. It's the sum total of all the biological reactions in the body. And the biochemical reactions in the living cells, we call them metabolisms. So all the biochemical reactions in the living cells are called metabolisms. And these biochemical reactions are organized into metabolic pathways. So here, I need to give one clarification. Generally, the biochemical reactions in the body are one. But because of how the complex it is, that is why it's split into pathways. Like for example, we have protein metabolism, carbohydrate metabolism, nucleic acid metabolism, and lipid metabolism. So all these metabolisms are interconnected with one another. They are interconnected with one another directly or indirectly. But because of the complexity of the reactions, that is why they are split into pathways. And each of these pathways have their own purposes. So for example, the pathways have dedicated purposes. Number one, some of the pathways are involved in the extraction of energy. Of course, we know that in the living system, we have some pathways that are involved in the extraction of energy. When we say extractions of energy, it means they are the pathways that usually produce energy in the living cells. An example of these pathways, we have glycolysis. Generally, in glycolysis, we extract ATP. In citric acid cycle, we also extract energy. In the beta oxidation, we also extract energy. Oxidative phosphorylation. So all these pathways are the energy generating process. And we have some pathways that are specifically for the storage of fuels. Like for example, if we take glycogenesis, that is the synthesis of glycogen. You know that generally when we take excess glucose, and the glucose taken provide the enough energy of a living cells in that particular time. So the remnant of that glucose will be converted to glycogen in the liver and store. So the process of converting glucose to glycogen is called glycogenesis and is one of the pathways that usually store fuels in the form of glycogen. And another examples we have like lipogenesis, that is a synthesis of lipids from acetyl-CoA in the adipose tissue. It's also a way of storing fuels, and we call it lipogenesis. And the synthesis of important building blocks, of course, we have like pentose phosphate pathway is one of the pathways that usually generate a building blocks which are used for some important synthesis of some biological materials. Like for example, in the pentose phosphate pathways, we usually generate pentose sugar. which serve as a building block for nucleotide synthesis. And of course, you know that these nucleotides are the building blocks of nucleic acid, DNA, and RNA. And so also we have some pathways that are involved in the elimination of waste materials. Like for example, you know that generally nitrogen is toxic in the body when it is excess. It causes a disease like hyperammonemia. And of course, we have a pathways that usually involve in the elimination of waste materials like urea cycle. So all the pathways that are splitted have their important or have their purposes. So that is why it is very, very important to split these pathways for better understanding. And the pathways can be represented as a map, of course. The pathways, we can usually represent them as a map. And these pathways follow the fates of metabolites and building blocks. So we know that if we are talking about a pathways, we must talk about the fates of the metabolites. Like for example, if we have glucose in glycolysis, the glucose will convert to glucose 6-phosphate, and the glucose 6-phosphate will now serve as a substrate for the next enzymes, which is which is involved in adding another 1-phosphate group, which is fructose 1,6 bisphosphate. Fructose 1,6 isomerase. So the substrate of one enzymes, so the product of one enzymes is usually served as a substrate of the next enzymes. So it follows the fates of the metabolite and the building blocks of the metabolism. Then also the metabolic maps identify the enzymes that act on these metabolites. And it also identify points and agents of regulation, and it also identifies sources of metabolic disease. So this is actually the main importance of the metabolic map. One, it will give us an idea of the metabolites of each of the particular feats of a pathway, and also the building blocks. It also helps in identifying the enzymes that act on these metabolites. So also at the same time, it also identifies the points and the agents that are involved in regulation, that is in the regulation and the control. When you say regulation, it means the control of the metabolic pathway. Of course, in each and every pathway, there is a control point. So this metabolic map will help us in identifying the points of regulation and the agents that are involved in the control of that pathway. And at the same time, it helps us in identifying the sources of the metabolic disease. Is it as a result of the increase in the activity of a particular enzyme that leads to the production, that lead to increase in the production of the products of that enzymes, which may lead to colossal accumulation of that, leading to a disease condition? Or is it as a result of the decrease in the activity of the enzymes, leading to insufficient production of the products, causing metabolic disease? So all these are important in the metabolic map. So now this is our metabolic map and the map of metabolic pathways. So you see that we have like glycogen, biosynthesis, and metabolism. This one is our metabolic pathways on its own. We have nucleotide metabolism, we have biodegradation of xenobiotics, we have carbohydrate metabolism, we have lipid metabolism, energy metabolism, biosynthesis of secondary metabolite, metabolism of cofactors and vitamins, amino acids. So all these are the metabolic pathways. And one thing that I want to each and everyone of us to understand. In a typical eukaryotic cell, it has the capacity to produce about 30,000 different proteins. And these 30,000 different proteins are involved in catalyzing thousands of different reactions. And these thousands of reactions are involved or involve hundreds of metabolites. And they must shared by more than one pathway. So, ladies and gentlemen, it is very important to understand that in living in a typically eukaryotic cell, there are over 30,000 different proteins. Some of these proteins are mostly enzymes, and these proteins are involved in catalyzing thousands of reactions. And these thousands of reactions are involved by hundreds of metabolites, and these hundreds of metabolites are linked with one pathway to another. So in much simplified overview of metabolic pathways, each dot here, each dot is representing an intermediate compound, and each of these connected lines represent an enzymatic reaction. So if you see that, you will see that we have a number of dots. So all these dots is representing an intermediate compound. And the connected line, it shows that there is enzymes reaction. It means that look at it from here to here, it means there is an enzymes reaction. So also, like for example, from here to here, it tells you that there is an enzymes reaction. So this is a connected lines. So for more realistic and for more complex diagram of metabolism, there is one online database we have. We call it KEGG pathway database. And in this, you can just take this map to that database, that is KEGG pathway. It will, in each of these connected line, it will tell you in the database, it will tell you which of the substrate is that and which of the enzymes is that. So it is very, very important. So now the next thing is the principles of regulation. So the flow of metabolites through the pathways is regulated to maintain homeostasis. So you know that this homeostasis, we can have homeostasis at different level. We can have homeostasis. We know that this homeostasis, we did actually homeostasis since from secondary school in biology. So it also here, we are also have it here in biochemistry. So homeostasis can be regulated at molecular level, at cellular level, at tissue, organs, and the systems levels. So we have homeostasis in all the level of organization of life. Because one thing that I want us to understand is if there is no homeostasis at molecular level, so it means that because any problem starting from molecular level, it will also be reflected as at the organism level, that is at the systems level. So if you have the problem of the homeostasis at molecular level, that problem will also manifest in the living organisms. So it is very, very important in homeostasis to maintain the concentrations of the metabolites to be kept at a steady state in the body. Because if it did not keep kept at a steady level, of course, it may lead to the risk condition. Like, for example, let me just give us an example of how um, homeostasis problem causes a disease condition. Let's say like in diabetes mellitus, for example, the regulation of blood glucose level is defective as a result of the lack of or insensitivity to insulin, which profound medical consequences. So you see, it's because we have the problem of insulin at the molecular level. That is why the glucose concentrations is not kept at a steady state. And as a result of that, we have a disease called diabetes. So that is why it is very, very important to understand how these homeostasis are maintained at molecular level. And so also when we perturbed reactions, so a new steady state is achieved. It means that whenever you alter or you change, or whenever body adjusts to a particular change in conditions, so a steady state will also, a new steady state will also be achieved, so that to make sure that the body is kept safe. So sometimes the levels of required metabolite must be altered very rapidly. So now what I want us to understand is this. To achieve homeostasis, always the number of the substrates that are utilized in one particular pathway must also be in steady states with the products that are produced. So as I say, sometimes the levels of required metabolite must be altered very rapidly. And why do we need this? We need this to increase the capacity of glycolysis during action. So there is actually some time that we have that as a result of the activities we are doing, there is a need to alter a particular pathway. Like for example, of course, if we need enough of energy, so there are some period, or there are times that the glycolysis metabolism must be altered so that to meet the demand of the body's cells. Like for example, needs to increase the capacity of glycolysis during action. And there may be a need to reduce the capacity of glycolysis after the action. Of course, if you are running, you are using a lot of energy. So at that point, the level of glycolysis have to be increased so that at least you will produce enough energy that will maintain, or that will help you to maintain that running, that action. And after the action, the glycolysis have to be reduced so that to maintain the body demand. And there are some times that need the capacity of gluconeogenesis, that is after successful action. Gluconeogenesis is the process that involves the synthesis of glucose from non-carbohydrate source. So of course, for example, if we run and we use a lot of energy, of course, a lot of building blocks will be produced. And of course, after running at that time, maybe you have a lot of the building blocks of gluconeogenesis. Of course, the building blocks will now be converted back to produce glucose, and the remnant of the glucose, if it is not needed at that time because it reduced after action, then it will now be converted back to glycogen. So feeds inhibition. In many cases, we need to understand that in many cases, the products of one metabolic pathways directly or indirectly inhibit their own biosynthetic pathways. Like for example, why do we have glycolysis? We have glycolysis because we need energy. So if you have excess of this ATP, do you need it anymore? No, you don't need it anymore. So therefore, the glycolysis have to stop. How does it stop? Then the high ATP produced will now inhibit the committed step of glycolysis so that to prevent excess glucose degradation. So this kind of inhibition, we call it feedback inhibition. That is the end product of a particular pathway will now come and inhibit the committed step. When we said committed step, it means that the point of regulation, like for example, in glycolysis, we have like 3 points of regulation. And the main enzymes that are involved in the regulation is fructose 1,6. I think yes, it's fructose 1,6 kinase enzymes, of course. So therefore, at that point, the ATP will now inhibit the enzymes, and once it is inhibited, there is no more excess glucose degradation. So now, the rate of biochemical reactions. So how do you measure the rate of biochemical reactions? You know that when we said the rate of biochemical reaction, it means to obtain the rate of biochemical reactions is based on disappearance of the substrates or appearance of the product per unit time. But we have to understand the rate of biochemical reaction, it depends on some factors. Number one, concentrations of the reactant versus the products. And number two, the activity of the catalyst. So sometimes if you want to increase the rate of the reaction, of course, you can look at it from increasing the concentration of the enzymes. So the rate of translation versus the rate of degradation. So in this point, what you are trying to say is if you want to increase the rate of the reaction, you can increase the rate of the reaction, one, by increasing the concentration of the enzyme. And how do you increase the rate? So yeah, how do you increase the concentration of the enzymes? You can increase the concentration of the enzymes by increase in the translation of that proteins, because enzymes are proteins, so they follow the same pattern as other proteins. That is the central dogma of molecular biology. And we know that the central dogma of molecular biology is started from replication, transcription, and translation. So if you need that enzymes in excess quantity to increase your rate of the reaction, so it means that you need to increase the rate of translation of the enzymes concerned. And if you want to decrease the rates of the biochemical reaction, of course, you can increase the rate of the degradation of that enzymes. Of course, we have some proteins or some enzymes that after they elicit their catalytic activities, they have to be degraded. So how do they degrade it? We have what you call proteosomes. Proteosomes are a group of proteins that usually involve in the degradations of proteins when they elicit their physiological role. Like for example, let's take example with adrenaline. We can't have adrenaline all the time in our body because it will make us to be excited, to be actually unstable. So we need only that that hormones adrenaline when it is needed. So when it is needed, the rate of the synthesis will be increased, so it will produce a large amount. And once it elicits its role, then it will now be degraded by the proteosomes. Then another thing, intrinsic activity of the enzymes. Of course, we can increase the activity of the enzymes, and this increasing the activity of the enzymes depend on some other factors. To increase the activity of the enzymes, you can also maybe increase the substrate concentration. If you increase the concentration of the substrate, of course, the enzymes may increase and the effectors as well, or maybe phosphorylation state of the enzymes. So So it means that if you want to increase or decrease the concentration, so the rate of the biochemical reactions, you can either increase the concentrations of the reactant or products, or you can increase the activity of the catalyst. So increasing the activity of the catalyst, it can be in the case of increasing the concentration of the enzymes or increasing the activity of the enzymes. So also another way of increasing the rate of the biochemical reaction, also increasing the concentrations of the effectors. So number one, increasing the concentration of the reactant versus product is one of the way of increasing or decreasing the rate of the biochemical reaction. Increasing the activity of the enzymes. So to increase the activity of the enzymes, we can look at it in two ways, either by increasing the concentration of the enzymes or increasing the activity of the enzymes. So for the increasing the activity of the enzymes, we can look at it at the increase in the rate of the translation of that particular enzymes or versus the degradation of that enzymes. And for the intrinsic activity of the enzymes, we can look at it based on the increase in the concentration of the substrate or decrease the effectors or the phosphorylation state of the enzymes. There are some enzymes, when they are phosphorylated, their activity will be increased, and when they are dephosphorylated, their activities may be decreased. So we can look at it in that way. Also, another three factors that we consider in determining the rate of biochemical reaction is the concentration of the effectors. So the concentration of the effectors, these effectors can be allosteric regulators or competing substrate, pH, or ionic environment. So these are the effectors that help in increase or decrease in the activity of the enzymes. There are enzymes that act maximally or that functions maximally at acidic pH or basic pH or neutral pH. So you have to look at it at that point. So assuming now you have an enzyme that usually act on acidic and you introduce that enzymes into basic environment, of course it will not work well. And another factors we can consider in increasing the rate of biochemical reaction is the temperature. So some enzymes functions or act maximally at room temperature or body temperature. So if you decrease the if you decrease the temperature of the environment, the enzymes may become inactive or the activity of the enzymes may be decreased. And if it is above that, it may become inactive or even denatured. So that is this. So these are the factors that determine the activity of the enzymes. So these are like a way of how the enzymes activity will be increased, either by the increase in the concentration of the enzymes or maybe increase in the activity of the enzymes. So of course, for we to have increase in the concentration of the enzyme, there must be a signals, an extracellular signals that will tell the body that we need these particular enzymes or proteins in large quantity. So you see, this is an extracellular signals, and these extracellular signals will now lead to activation of some hormones, maybe like adrenaline. Of course, like for example, let's take example with adrenaline. Adrenaline is a hormones that usually involve fight, and anxiety hormones. So when you are excited, of course, you will feel that you have enough energy, or if something makes you to fear, like for example, if you are in the classroom and you just saw a snake coming into the class, of course you will be excited. And what makes you that excited is because of the adrenaline, because the adrenaline makes you to have enough proteins that will make you to feel that you have an energy that you can even jump from an unexpected building. Okay, so that is it. So assuming now these extracellular signals trigger the expression or trigger the production of adrenaline, and this adrenaline now will bind to the receptor, and this receptor with some will now trigger the activation of transcriptional factors, and these transcriptional factors will now enter into the nucleus of the cell. And in the nucleus of the cell, these transcriptional factors, look at them here, will now bind to the promoter region, or it will now bind to the DNA, and the DNA from there, it will now bind to the promoter region to start synthesizing messenger RNA, and this messenger RNA will now further be undergo transcriptional modification, and at the end, the M messenger RNA will now actually exit from the nucleus, and once it is outside the nucleus, introns and exons are in the messenger RNA, and it will undergo splicing. Then from there we'll have mature messenger RNA. The mature messenger RNA will now come to the ribosomes where it attached to start synthesizing the protein. So of course, this is the proteins, it means that this is your enzymes. And of course, if you only need, if this messenger RNA maybe finish or elicit its role, it will of course undergo degradation. So after these proteins, which is enzymes, is produced, then of course it will now start its role. Maybe let's say this is the enzymes. So of course the enzymes can undergo a number of modification, and look at it. If the proteins is produced, let's say this is the enzyme. If the enzyme is produced and it's finished its role, of course, as I said, it will undergo degradation, protein degradation through ubiquitin proteasome activation. So, and from there, the enzymes, for example, enzymes sequestered in subcellular organelles. So it depends. There are some enzymes, when they are produced, they will be actually stored, and usually is occur in the endoplasmic reticulum. So once it is stored, it will sequestered and store. And number 7, enzymes can bind to substrates to produce a product. That is another way. Then at the same time, these enzymes can also bind to ligand. A ligand, look at it, this is a ligand that binds to the enzymes, and we call it allosteric effector. So, and this is also enzymes undergo. There are some enzymes that do not actually act, that they are not active until when they are phosphorylated. So this is how they are now phosphorylated. And there are also some enzymes that they do not work until they combine with regulatory protein. So all this is just to give us an idea of how the enzyme's activity can be increased. Okay. So now the rate of the chemical reaction depends on the concentration of the substrate. So I think, ladies and gentlemen, I can stop this lecture here. Then we will continue next time. Thank you.